discovery of new site. So before I'll start with pathway specific approach methods used to activate silent bisynthetic gene cluster. I will discuss about katong sa box 1 ma'am and box 2 box 1 and in box 1 kay it's about the two alternative methods to identify new to activate silent bisynthetic gene clusters. So the first one is about HTS. So HTS identify target proteins such as establishing that a gene is important for survival. So the protein is then screened with compounds that might serve as potential inhibitors. So the reason nga nung gamitan siya potential inhibitors kay because kailangan man ma-identify ang target protein kailangan ma-identify ang ang property sa ka target protein. So if ever nga if na known nga kaan hanang inhibitor nga mo interact sa kaan sa maong mystery protein kay muna siya dito may balan nga nganong kaan kanang unsa to nga target protein. So that pero napud ni siya yung mga kuan limitation sa kuan in using HTS. So kanang the first limitation sa HTS kay kuan siya kanang ang process sa ang ni-occur inside the cell kay kuan kanang mo result siya o kanang dili same process sa ni-occur sa inside the cell. So if kaning HTS ang naay possibility nga ang process nga ni-occur sa outside sa cell kay dili siya same sa ni-occur sa inside nga cell. So the
Okay,
next
so in
is
short, of course, or this method is done outside the context of the cell. Okay, so uh, um, different factors, environmental factors, might also change the expected output. Okay, next, Miss Tubir.
then and another limitation is kani pong HTS kay kuan pod mas less yang affinity for kuan specific target protein. Then next is katong the second kuan ma'am kay second um methods to alternative methods used kay kuan katong fragment based design. Kani fragment based design involves the use of chemical fragment with a low binding affinity to the target. Then this kaan this fragment based design kay mas advantage siya sa compare sa HTS kay mas kuan hanang experimental cost siya and kanang maka develop siya multiple ways to develop novel compounds no. Then katong sa box 2 ma'am kay about man to siya og kaan kanang mga types of metabolite ma'am. Then kung unsa nga enzyme ang mo synthesis ato nga unsa nga enzyme ang mo synthesis niya like katong sa mga sa atong enzyme nga natin is polyketides which is katong from the decarboxylation condensation of one or more alkyl. So kuan ni siya, then di anang uh, kani nga polyketides kay i-synthesize na siya PKS ma'am, atong enzyme nga PKS nga which is a multi-enzyme nga maka synthesis of polyketides. Then so next is kaning about nito sa approaches. The next approach katong 'di ba I discussed last meeting about pleiotropic approach. The next approach or methods to activate biosynthetic gene cluster is the pathway specific approach. So the first method under pathway specific method is manipulating pathway specific regulators. So advantages of this method is it allows precise controlled activation of a site specific biosynthetic gene clusters. However, there are limitations in using manipulating pathway specific regulators. So the limitations requires that the BGC iteras be controlled by an identifiable transcription factor that is genetic manipulation. This is an example of compounds that are being developed using manipulating pathway specific regulators. The next method under pathway specific method is the reporter guide mutant selection. The advantages of this is combined combines a pleiotropic mutagenesis strategies with a pathway specific detection method for visualizing mutants expressing the biosynthetic gene clusters of interest. However, the limitation of this method is it requires cloning of promoter sequence from a target biosynthetic gene clusters. So example of compound being developed using reporter guide method under pathway specific approaches. So next is lastly refactoring. So the advantages of this is affords precisely controlled activation of the pathway of interest. The limitation of this method is natural promoters must be readily identified and BGC must be amenable to genetic manipulation. Lastly, so heterologous expression. So the advantages of this is simply the identification of metabolites. Then the limitation is that traditional methods for heterologous expressions are limited to small gene clusters. So, dili siya pwede sa kuan ka ng dako nga big og sa di siya pwede sa ng mga large gene clusters, ma'am. Kay limited to kuan naman siya, small. So, example of compounds being developed is the avermetolol. Then, kani siya figure for as an example of pathway-specific strategy that have been used to induce the expressions of silent biosynthetic gene clusters. So, the first is kani siya kabahin og manipulation of pathway specific. The first A and B is example of manipulation of pathway specific regulatory genes. So, kani siya kay kaning mga activator gene and kaning repressor gene kay kaani siya ma'am sila mo ga controlled sa specialized metabolites sa biosynthetic gene clusters. So, kani nga is used to activate silent biosynthetic gene clusters. However, kaning mga activator, ang natural promoters, which is the activator genes, kay islan siya og uh, constitutive promoter to activate silent biosynthetic gene class to activate biosynthetic gene clusters. So kani nga kaan example ani kay I compare aning sa B kay ang iya kay deletion of repressor gene. So kaning 'di ba ang repressor gene kay siya mo use to activate kuan silent by activate biosynthetic gene cluster. Kaning sa deletion kay naay mga kuan gene nga kailangan i-delete to kanang kailangan i-delete to activate kuan biosynthetic gene cluster. So kani nga gene kay moag di ba repressor gene, so aning nga sa deletion of repressor gene kay i-delete ang naay ubang gene sa example katong sa SCRBR2 kay na siya i-encode na siya mo encode na siya a repressor of CPKBGC in streptomyces color. So if ang if i-delete siya so that maka-cause siya og production of katong yellow pigment sa kakuan. So specific kuan siya if ang sa tawag ana mo undergo sa deletion sa of repressor gene so that maka-activate siya og biosynthetic gene clusters, then maka-produce siya og product. Then kaning sa C is
So the repressor gene there functions for inhibiting expression of the biosynthetic gene cluster. Therefore, in that method,
so therefore
okay, go on.
ma'am kaning deletion kay ang repressor gene kay iyang i-delete so that maka ipuli ba to para maka kuan siya lain nga production to activate
Naiipuli or deletion of the repressor gene will allow expression of the biosynthetic gene cluster.
oh yes ma'am.
Okay, go on.
Then sa C kay next is the kani siyang pathway specific strategies kay use niya kay refactoring and or heterologous expression. So kani siya kay ang constitutive promoter kay kaan siya ma'am kanang exchange sa silent BGC to gi-exchange siya ani ma'am kay para gi-exchange siya to activate silent BGC. Then kaning Victor ma'am kay pero kani manggong sa refactoring ma'am kay kuan na siya kanang kailangan na siya og kanang dili kay siya handle og kanang mga large gene clusters ma'am. So gamitan siya og Victor kani to para maka kuan kanang maka capable na sila to kuan to kanang to carry large gene clusters ma'am. So the next is methods for metabolite identification. So kani they in this article they use high pressure liquid chromatography. So kani siyang example kay
Also known as
ay wait.
HPLC. By the way, guys, the HPLC is available at the chemistry department of NORSO. So if you want to do thesis in the future in which you want to use, there is a need to use HPLC. It's actually available at the chemistry department. Okay. So this is usually used to separate metabolites. So if you want to study effects of metabolites, they usually, diba, in some researches, they separate metabolites in order to check for any efficacy. For example, kuan ba siya? Teka, gani. O pwede ba siyang herbicide or pwede ba yung antimicrobial or antibacterial, antifungal, or it prevents production of blood vessels. Okay, so nataana sa chemistry department. I don't know if the chemistry department will allow you to borrow the equipment. But anyways, available. Okay. So that's the main function of HPLC. Sige. Go on, Miss Bill.
So then sorry. Kaning sa solvent kaning solvent mobile mobile phase reservoir kay kuan ni siya usually a mixture of polar and non-polar liquid components. Then kani siyang pump solvent, manager solvent kay kani siya mo paingon siya sa mo for sa solvent nga mo paingon sa system color and detector, which kaning detector is mo detect siya sa analyze kanang mga ilang gi-extract from kaning detector ma'am kay mo ni siya mo detect sa kuan kanang mga sample nga extract gikan sa kuan gikan aning
Are you sure about that, Miss Tubil? I think you're you're jumbling words.
oh,
You start
ay
first
sorry
with the
ma'am. Wait.
solvent. It's okay, it's okay. You start first with the
Oh,
solvent reservoir, then what's next?
pump solvent ma'am.
Then what happens there?
Kaning sa pump solvent ma'am kay kuan siya um gikan ay iyang gipump ning mga gikan solvent mobile phase reservoir iyang gipump paingon sa uh, kaning kuan ma'am iyang gipump paingon na ning wait. Iya ipump to system columns for kuan kanang ay chak iya ipump to paingon diri sa high performance chromatography column ang gikan di ba gikan sa solvent ma'am. So ang kaning pump solvent siya mo pump so that ang magikan diri mo paingon sa chromatography
Okay, Miss Tubil,
column.
instead of
Then
explaining it
kaning
to me, excuse me, Miss Tubil, instead of explaining it to me, please explain it to your classmates instead. Huh?
ah sorry ma'am. So kaning solvent ay kaning pump solvent manager solvent delivery system kaya siya may mo pump sa mga solvent to chromatography column. Then this kaning chromatography column kay ani siya kanang gikan diri mo paingon siya i-detect siya detector kay kung unsa tong mga na extra nga sample diri or material i-detect siya diri sa detector. Ang detector may ma-detect sa mga solvent nga napaingon diri sa chromatography column. Then this kaning computer data kay siya mo'y mga integrate og response nga na-detect sa detector nga nang like iyang gi-interpret ang data nga na-detect sa detector.
What does the fixed
So
theorem
the
mean?
pardon ma'am?
What does the fixed there mean? Anyway, that's for organic chemistry. That's already in chemistry. Continue na lang. Those are instrument
Sige
data.
ma'am.
Can you
So
move on
kanish
to the next, Miss Koan? Miss Bill, move on na lang sa next.
dili na lang ning apelo ning kaan ma'am
Yeah,
and
let me see the
and
next slide.
and MR.
Yes, let me see the next slide.
Kuan na pud ni ma'am katong X-ray.
How
Then
about
summary na ma'am.
itong sa article? Bioinformatics, did you include that?
Sa asa ma'am?
Itong last figure, I think that was the last figure, in which first the different strategies followed
Katong
by,
last
yes, did you include that in your presentation?
katong last kuan ma'am?
Yeah, the last, I think
Because
that
year
was the
2?
last one.
Ay, kay
No,
ang
the
last
bio,
figure ma'am kay figure 4 kay katura man siyang
okay, let me see the article. What ni mo gi-appeal, Miss Ruby?
asa
Whenever you report,
katong sa
include all the
figure 2 ma'am.
figures. Let me check for a while. Yeah, figure 2. Did you include that already?
Murag wala ko
Figure,
ka include ano ma'am.
wala man gi-include niya. Ni-include ka din nag-HPLC o NMR.
Ay,
Hmm. Skip ano mo. Ay na lang to, Miss Tubil. Anyway, most of you have read the article, so I assume you have read that already. Okay, let's move on to the next reporter.
thank you ma'am.
Morning. Maklaro na ko, ma'am.
Maklaro ra ka, Miss Abril. Maklaro yung presentation, Miss Abril.
Ha?
By the way, can you rotate your screen to
I-landscape ti Abril.
Wait. Kaya ako naman yung GJP. Good. May. Wait. Kung na JP, good ko. Okay na?
Kana, better.
Ako sa set up. Okay. Good morning, ma'am. Good morning, classmate. Okay. Today I will discuss one type of cell division, which is mitosis, or known as mitotic phase. Okay. This is the outline of my report or topic. Okay. Introduction. A human, as well as sexually reproducing organisms, begins life as a fertilized egg or zygote. Trillions of cell divisions subsequently occur in a controlled manner to produce
Hello, hello, hello. Nana po ng inyong presentation nga. Puros text. Diba I told you, i-bullet siya and just include the salient points and then explain
Okay.
the rest of the sentences
Next time.
instead of placing them there.
Wala.
Anan inyong kapot. Wala na ko gabalik-balik og ingon.
Okay,
Okay?
ma'am. Okay. Trillions of cell divisions subsequently occur in a controlled manner to produce complex multicellular human. In other words, that original single cell is the ancestor of every other cell in our body. Once a being is fully grown, cell reproduction is still necessary to repair and regenerate tissues because all multicellular organisms use cell division for growth and maintenance, repair of cells and tissues. Like for example, our blood cells, our skin cells, they are cells that are constantly being produced in our body. Cell division is tightly regulated because the occasional failure of regulation can have life-threatening consequences. Okay, if there is a failure of regulation during cell division, there is an error or that would kanang result into an error or a mistake. Okay, one cause of that mistake is the inactivation of tumor suppressor gene or P53. Okay, these tumor suppressor genes, these are normal genes that slow down cell division, DNA repair DNA mistakes, or tell cell when to die. And when this tumor suppressor gene don't work properly, the cell can grow out of control, which can lead to cancer.
Miss Adele, you can include those molecules once you start discussing interface.
Ma'am,
You can include those molecules once you start discussing interface. It's not on your PowerPoint presentation. Did you include that in the interface section of your report?
wala ma'am. Molecule?
Specifically,
Ma'am?
nag-isip na tumor suppressor gene. Did you include cyclin, dependent kinase, katong iyahang buan, cycle?
Wala.
Sige, para yun sa
Okay. Single-celled organisms use cell division as their method of reproduction. Okay, one example is bacteria. They use cell division as a method of the reproduction. For example, is binary fission, the asexual reproduction of the bacteria. While there are a few cells, um, wait. While there are a few cells in the body that don't undergo cell division, like for example, the sperm and our egg cell. Okay. What is cell division? Cell division is the process by which a parent cell divides into two or more daughter cells, involving both nuclear and cytoplasmic division. Cell division usually occurs as part of a larger cell cycle. There are two types of cell division: mitosis and meiosis. Mitosis is a process where a single cell divides into two identical daughter cells. This is most this is used by our somatic cells in our body. While meiosis is a process where a single cell divides twice to produce four cells containing half the original amount of genetic information from the original cell. Wala, nagkabaylo. Kanidi. The mitotic phase alternate with interphase in the cell cycle. Okay. In 1882, there is a German anatomist named Walter Fleming developed dyes that allowed him to observe for the first time the behavior of chromosomes during mitosis and cytokinesis. During the period between one cell division and the next, it appeared to Fleming that the cell was simply growing larger. Okay. Walter Fleming, who also coined the term mitosis and chromatin. Okay. Phases of the cell cycle. Okay. There are two phases of the cell cycle.
By the way, hello, Miss
Ma'am?
Abril, before you continue,
Yes.
can you differentiate chromosome, chromatin, and chromatid?
Oh, wait, ma'am. Chromos. Okay, chromosome, this is a long DNA molecule with part of all the genetic material of an organisms, while chromatin, it is a complex of DNA and proteins that forms chromosome within the nucleus of eukaryotic cells.
So how are they different?
Okay, this chromatin, this is a thread-like structure that will form into chromosome when it is condensed.
Okay, how about chromatid?
Chromatin, chromatids, ma'am. Okay, this is two sister. Okay, this is two sister chromatids that form chromosome.
When are you going to use chromatin? When are you going to use chromosome?
Chromatin. Okay, chromatin is formed before kanang before it condense in or it will kanang before pa siya mo form into chromosome, ma'am, kanang before the process of kanang cell cycle begin.
Asa before?
Before cell division begin.
Okay, at what particular stage? Can you be more specific, Miss Abril? At what particular stage of
During
cell division?
interphase.
Okay, please proceed to your report.
Okay. Cell cycle has two phases, the interphase and mitotic or known as M phase. Okay, interphase, this is often account about 90% of cell cycle, and however the mitosis or the M phase is the shorter is usually the shortest part of the cell cycle. Okay, interphase subdivided in two subphases. The G1 or the first gap, followed by the S phase or the DNA synthesis, and the G2 phase or the second gap. Then in G1 phase or the first gap, referred to as the growth phase. Okay. This is the time which the cell grows by producing proteins and cytoplasmic organelles, such as mitochondria and endoplasmic reticulum, ribosomes, lysosomes, and Golgi apparatus. Okay. During this phase, the cell synthesizes various enzymes and nutrients that are needed later for DNA replication and cell division. Next is the S phase or the synthesis phase.
Wala kay slide para ana, Miss Abril.
Kanira ma'am nga figure.
Okay.
Okay ra di, ma'am?
Ang mura man dagway na sige ka na lang.
Sige, ma'am. In its phase or the synthesis phase, the cell synthesizes a complete copy of DNA in its nucleus. It also duplicates a microtubule organizing structure called centrosome. Okay, the centrosomes help separate DNA during M phase. Okay, this organelles, this centrosome is only present in animal cell, not in plant cell. Okay, and the last phase is the G2 phase or the second gap. Okay, the last part of interphase. The cell has grown, DNA has been replicated, and now the cell is almost ready to divide. This last stage is all about prepping the cell for mitosis or meiosis. And then, next is for the mitosis, conventionally broken down into 5 stages: prophase, prometaphase, metaphase, anaphase, and the telophase. Overlapping with the latter stage of mitosis is the cytokinesis. Cytokinesis completes the mitotic phase. Mitosis in an animal cell. Okay. As you can see in the figure, in the upper part, that is an animal cell under the microscope. Okay. Okay. In the G2 of interphase, okay, a nuclear envelope encloses the nucleus. Next is the nucleus contain one or more nucleoli, or in singular is nucleolus. Then next, two centrosomes have formed by duplication of a single centrosome. Centrosomes are regions in animal cells that organize microtubules of the spindle. Okay, each of centrosome contains 2 centrioles. Next is the chromosome duplicated during S phase cannot be seen individually because they have not yet condensed. Okay. Okay, the first phase in the mitotic phase or mitosis is the prophase. The chromatin fibers become more tightly coiled, condensing into discrete chromosomes observable with a light microscope. The nucleoli disappeared. Each duplicated chromosome appears as 2 identical sister chromatids, joined their centromeres, and in some species, all along their arms by cohescence or sister chromatid cohesion. Next is the mitotic spindle begins to form. It is composed of centrosomes and the microtubules that extend from them. Okay. Okay, the radial arrays of shorter microtubules that extend from the centrosomes is called asters.
Can you discuss the 3 types of spindle microtubules, Miss Abril?
Spindle microtubules. Wait lang, ma'am. Spindle microtubules. Wait sa, ma'am. Wala ko kakuha na, ma'am. Wala na.
Nya, naman na subok.
Wait lang. Huli. Mm. Kanang, wait lang, mama. Microtubules. Wala ko ka. Wait lang, mama. Microtubules. Okay. The three kinds of microtubules are the kinetochore microtubules, the astral microtubules, and the spindle microtubules. Kinetochore microtubules, this is where the kinetochore of the chromosomes is attached to, while the astral microtubules, this is where the kanang spindle fibers is attached, ma'am.
Ito to, ang spindle microtubule.
Ano?
Spindle fiber. Mitotic spindle.
Dali, wait. Sige pa. The kinetochore microtubules, this is kuan kanang where the kinetochore of a chromosome attached. Okay, the non-kinetochore microtubules, these are the spindle fibers. Ano ba? Asa mo na? Wait lang, hala. Wait. Hello, ma'am.
Figure out kanimo?
Okay. Ang non-kinetochore microtubules, ma'am, this is where kuan kanang wala siya na attach sa kinetochore of each chromosomes.
Asa siya naka-attach?
Wala na attach nga kuan chromosomes. While the kinetochore microtubules,
First, the
These are
difference.
microtubules that where the kinetochore of the chromosome of each chromosomes in the cell is attached.
Kindly open your kuan sa pag. Can you encode it in your PowerPoint presentation so that others can also see?
Ma'am?
Include sa imong PowerPoint presentation.
Wait. Na diri na separate ag usa nga kuan, ma'am. Kana ra.
No, the three types. Can you enumerate it and write it on your PowerPoint presentation? Maka-add pa kag slide in, ha?
Dili kay JPEG ra man ni ma'am akong gikuan. Sa cellphone ra ko gakuan kay ang laptop loading man kaayo.
Okay, kindly enumerate the different spindle microtubules first before you continue.
Okay. The three distinct types of microtubule fibers is kinetochore microtubules, the polar microtubules, and the aster microtubules.
So what's the function of each of those?
Okay, kinetochore microtubules. These microtubules hold the chromosomes, the kinetochore of the chromosomes were attached in these microtubules. While the aster microtubules, this is where the spindle fiber attached in both poles. Or then the kuan kaning non-kinetochore microtubules. This is a microtubule where kanang wala around to naka-attach ang kuan kanang chromosomes, ma'am. That is why it is non-kinetochore microtubules.
Okay, continue.
Okay, in the prophase. Okay. And the last process in the prophase is the centrosome. Centrosomes move away from each other, propelled partly by the lengthening microtubules between them. Then next is the prometaphase. Okay. In prometaphase, the nuclear envelope fragments, okay, or become fragments. The microtubules extending from each centrosome can now invade the nuclear area. As you can see in the picture, the spindle fiber is moved in the nuclear area in the center. Okay. Next is the chromosomes have become even more condensed. Okay. Naklaro na ang chromosomes ni condensed na siya. And then each of the two chromatids of each chromosome now has a kinetochore, a specialized protein structure at the centromere. Some of microtubules attached to these kinetochores, becoming kinetochore microtubules, which jerk the chromosomes back and forth. Okay. However, the non-kinetochore microtubules interact with those from opposite pole of the spindle. Next phase is the metaphase. Okay. In metaphase, the centrosomes are now at opposite poles of the cell. Okay. The chromosomes have all arrived at the metaphase plate. This metaphase plate is a plane that is equidistant between the spindle's two poles. The chromosome centromeres lies at the metaphase plate. As you can see in the arrow picture. Okay.
How are the chromosomes, Miss Brill?
Yes, ma'am.
Miss Abril, how are
Yes.
the chromosomes maintained at the metaphase plate or at the equatorial plate?
Maintain. Okay, they are maintained through through the kinetochore microtubules that hold the chromosome in place, ma'am.
Yeah, but how are they maintained in the middle of the plane? Kanang sa kuan nimo, imuhang equatorial plate.
Through metaphase plate.
Are you sure?
Oh.
How about the others?
Mm,
How about the others? Giyon sa pag-maintain nga naaras tunga ang imuhang chromosomes at the metaphase plate nganong naka-align man na siya. How is the alignment maintained?
through handling.
You have idea?
Wala, ma'am. Wala ko kakuan.
Mr. Gador?
How is the
What is responsible for maintaining the chromosome at the metaphase plate, or what is responsible for maintaining the alignment of chromosomes at the metaphase plate?
Ang microtubules, ma'am, from both poles of the mitotic spindle.
What about the microtubules from both poles?
Di ba siya kuan, ma'am? Nang siya specific nga length sa kuan. So, every ang singalan ani ang microtubules from like sa pikas nga poles ug sa pikas nga poles is the same measurement. So, mo siya ga-hold sa chromosomes nga mabutang siya nga naa sa metaphase plate.
Are you
Align.
sure it's about the length of the microtubule?
Ito ko idea.
How about the others? Mr. Banagodos?
centromeres.
Mr. Banagodos? What maintains the alignment of the chromosomes at the metaphase plate? What about you, Mr. Montegrejo?
Ano ma'am, it's the tug of war of some sort that occurs between the two spindle fibers that is attached to the chromosome.
Come again?
Ano ma'am, it's the force, it's the pulling and pushing that occurs, the pulling and pushing that the spindle fibers act on the on the chromosome. Katong magbirahan na sila ba? The tug of
Yes.
war.
Okay. It must be equal at both poles. Okay? So the forces applied on both poles should be equal in order to maintain the alignment of the chromosomes in the metaphase plate. Okay?
Okay.
Next question. Miss Abril,
Yes, ma'am.
why should the chromosomes be aligned at the metaphase plate?
So that during cy- so that during cytokinesis, the each of the daughter cells will have kanang the same number of chromosomes in each cell, ma'am.
Okay, go on.
Then the last process in the metaphase, okay, each chromosome, the kinetochores of the sister chromatids are attached to kinetochore microtubules coming from the opposite poles. Then go on to the next phase, which is the anaphase. The anaphase is the shortest stage of mitosis, often lasting only a few minutes. Okay. An anaphase begins when the cohescence proteins are cleaved. Okay. This allows the two sister chromatids of each pair to part suddenly. Okay. Each chromatid thus becomes a full-fledged chromosome.
How does these microtubules shorten?
Okay. It is shortened through the kuan. Kanang katura po Japong force, ma'am.
Are you sure?
Wait, ma'am.
Please answer my question, Miss Elnas.
10 because ay anya na ma'am. Dara day. Okay, this the kinetochore microtubule shortened because these microtubule are attached at the centrosome region. And the chromosomes move to centromere first at
Are you
about
sure?
micrometer in minute.
How does the microtubule shorten? Miss Elnas?
It shortens when kanang ang microtubules mo-attach na siya sa kanang captured nga kinetochore sa chromatin.
How does the microtubules shorten? Yes, Mr. Banagodos. Okay. All right. So there will be disintegration of the microtubules. Okay, that's why the microtubules shorten
Then,
during anaphase. Next.
okay. The next process, the cell elongates as the non-kinetochore microtubules lengthen. Okay, by the end of anaphase, the two ends of the cell have equivalent and complete collection of chromosomes. Okay, and the last phase in mitosis is the telophase. Okay, two daughter nuclei form in the cell. Nuclear envelopes arise from the fragments of the parent cells. Nuclear envelope and other portion of the endomembrane system. And also the nuclei reappear. Then the chromosomes become less condensed. Any remaining spindle microtubules are depolarized or depolymerized. Then mitosis, the cell division of one nucleus into two genetically identical nuclei, is now complete. That is where the cytokinesis will occur, ma'am.
Okay, what determines the position of the invagination or formation of the cleavage furrow, Miss Abril?
So,
What determines the position of the cleavage furrow?
and the what determines it? Kanang kuan ma'am. Sana i the two daughter cells is nearly separate or is nearly separated
Please
or
answer my question, Miss Triplet.
say to class.
Yes, ma'am. It is determined by the contractile ring of microfilaments formed in the cleavage furrow.
Okay, what determines the formation or the direction of the formation of the contractile rings? Remember, we're talking about animal cells, huh?
She's into
What determines the direction of the formation of the cleavage furrow? Diba, if you can still remember in the discussion sa microbiology, na atay kukos, unya ang kukos pwede chain, pwede cluster. Pwede siya grape-like, staphylococcus, o chain-like streptococcus. What is responsible? The one that is responsible for the formation of the chain is the direction of the cleavage furrow. So, ang direction of the cleavage furrow is vertical. However, ang katong sa staphylococcus, lain-lain ang iyang direction mo itong mag-cluster ang imong staphylococcus. Na ay horizontal, na ay vertical, na ay paslant. Okay. Now, there is also what is responsible for determining the position of the cleavage furrow is the position of the position of what? Remember how we're talking about
This plate,
animal cells. Yes, Mr. Motigrejo?
Please, ma'am.
Okay.
face plate.
The direction of the metaphase plate. Look, the metaphase plate, di ba horizontal? Therefore, ang iyang formation of the cleavage furrow is also horizontal. Kung paslant ng imuhang metaphase plate, paslant po ng cleavage furrow. In contrast to plant cell, lahi po ang sa plant cell, okay? Anyway, how about the checkpoints, Miss Abril?
uh,
Another question: what happens if cytokinesis does not take place? What will happen to the cell?
like it will form kuan ma'am cluster
Cluster of what?
of cells.
Unsa?
Wait.
Nahimo siyang multinucleated, no?
Okay, otherwise known as
Katong aneuploidy.
hoy 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 unsay aneuploide? Unsa man ang aneuploide? Miss Abril, unsa nang kuan? Unsa nang aneuploide?
Wait.
Utangan ka na ako, ana, Miss Abril ha, og checkpoints.
Yes, ma'am.
Hmm.
Ang chief.
Hasta tong kuan, katong CDK, cyclin dependent kinases, tong regulation.
Yes, ma'am.
Yeah, appeal to Miss Abril.
CDK. Yes, ma'am.
Okay. Thank you, Miss Abril. By the way, I forgot to thank you, Miss Tubil, for the presentation earlier. Thank you as well. Let's proceed to the next reporter.
Yes, ma'am. Thank you.
Ay, taysa, Miss Abril, pa dito imong plant cell. Di ba imo tong gi-appeal?
Yes, the last part.
Sige, present.
Sige, wait. Okay. The mitosis in a plant cell. Okay. The first phase in plant cell is this is the prophase, similar to the animal cell. Okay. The chromosomes are condensing and the nucleus is beginning to disappear, although not yet visible in the micrograph. The mitotic spindle is starting to form. And then in the second phase, in the prometaphase, discrete chromosomes are now visible. Each consists of two aligned identical sister chromatids. Later, in prometaphase, the nuclear envelope will become fragment. In the third phase, the metaphase, the spindle is complete and the chromosomes attached.
Miss Abril,
Yes,
rather
ma'am.
than discussing each of those stages again, please directly discuss the difference between mitosis in animal cell versus in plant cell.
Okay. Okay, animal and plant cell is uh, mostly similar in the process during cell division. However, there are kanang difference in the formation of during the cytokinesis occur kanang naa sila sa animals sa plant cell na ay cell plate nga mo appear in the telophase or the last phase, ma'am, while in the animal cells katong cleavage furrow.
Okay. If the position of the metaphase plate determines the direction of the cleavage furrow in animal cell mitosis, what about in plant cell mitosis? What determines the formation or the direction of the formation of the cell plate?
The position of the kanang cell plate, ma'am.
No gani. What determines the direction?
Similar to kuan katong there lang, ma'am.
I have discussed that in botany. Banagodos. Okay. What else? Sa pay difference nila sa
animal cell mitosis, Miss
Ang
Abril?
ang plant cell don't have centrosomes and centrioles
Okay.
where the spindle fiber attach in each pool.
Okay, what else?
They don't also have kanang
Why do they form cell plate instead of cleavage furrow?
wait lang. Ba- Maybe to kanang remain the I think kuan ma'am kanang kanang remain the two daughter nucleus separated. That is why they form a cell plate between the two daughter cells.
That's your next assignment, Miss Abril.
Yes, ma'am.
Why do they form cell plate instead of cleavage furrow? So first, I know not that's the third day. First, again, to reiterate, first, the assignment is to discuss the checkpoints.
Yes, ma'am.
Okay. And
Okay.
there are follow-up questions for each of the checkpoints, mind you.
Yes,
You have to
ma'am.
study it well. Second, regulation. Third, na may question a while ago.
Yes, ma'am.
Okay. Any questions from the listeners before we continue to the next reporter? Lara. Okay. Thank you, Miss Abril. Who's
Thank
next?
you, ma'am.
Who's next?
Ivy, ma'am.
Okay, sige. Mr. Lapa, you may start.
So, good afternoon, classmates. Good afternoon, ma'am. Now I'm going to report about meiosis and its processes. Okay, first, meiosis is the process where a single cell is divides into twice, produce four cells containing half the original amount of genetic information, and also meiosis is these cells are sex cells, from sperm in males and egg cell in females. During meiosis, one cell divides twice from four daughter cells. And the importance of meiosis is that because it ensures that all organisms produce via sexual reproductional contain correct numbers of chromosomes. Meiosis also produce genetic variation by way of the process of genetic recombination. So the person to discover meiosis is zoologist Oscar Hertwig. Meiosis was discovered and described for the first time in the sea urchin eggs in 1876 by the German biologist Oscar Hertwig. It is described in 1983 at the level of chromosomes by the Belgian zoologist Edward van Beneden and Ascaris roundworm eggs. Okay. So before we begin the meiosis, it undergoes the G1 or the root of the cell, and S phase where the synthesis of DNA chromosomes, and the G2 gap or the gap 2, then it begins into meiosis. Uh, during synthesis phase, the chromosomes or the DNA is being duplicated, and also the centrioles are duplicated. So that is during interphase or the synthesis phase. So meiosis, there's two types of meiosis: the meiosis 1 and the meiosis 2. After interphase, then the next is the prophase, where after the duplication of the centrioles and the the duplication of centrioles and the disintegration of the chromosomes, the spindle fiber will travel into the opposite pole, and the formation of the homologous pair of chromosomes will form. That is what we call synapses. Uh, Synapses or bivalent or a tetrad, where the homologous chromosome formed. And during this,
Can you discuss the detailed process of synapses and crossing over
yes, ma'am.
at the molecular level? Did you include it?
No, ma'am. Wala na ko sa part.
Okay, that's the first assignment.
Sige, ma'am. During synapses, the crossing over happened. Crossing over, it is the exchange of uh, genetic, though it's same, the exchange of genetic kuan genetic matters, pero they are not, though pare-pareha sila, but they are not exactly similar, so it would cause a kuan genetic variation. So, and after prophase, the metaphase the metaphase would occur, and that's the metaphase 1. Then after the spindle fibers travel, after spindle fibers uh, from the opposite pole, the spindle fibers would attach into the chromosome. The attach of the the spindle fibers will attach into the kinetochore of the chromosomes, the area ang kinetochore. So the difference between meiosis and mitosis, another difference sa kuan ay duwa na ako na discuss, another difference dai during prophase 1 dai sa meiosis and mitosis is that in meiosis there are homologous pairs of chromosomes, which is wala siya sa mitosis. That's the another difference sa meiosis o sa mitosis. And also during metaphase 2, the formation of the homologous na sila sa into the metaphase plate. So the
Okay, Mr. Salapa, to make it short, can you compare mitosis prophase versus meiosis 1 prophase, then metaphase, anaphase, and so on and so forth?
okay, during sa in meiosis during prophase 1, the formation of homologous pair is mahitabo siya in prophase 1, while in mitosis kay wala man siya homologous pair, so another difference na siya sa kuan.
So, unsa sa mitosis walay homologous?
Ay, yes ma'am, wala siya homologous pair sa mitosis, ma'am.
Do you agree with the statement of Salapa, Mr. Soliera?
The exchange of ang kuan day ang crossing over day, ma'am, wala sa mitosis nya mahitabo siya in meiosis during prophase 1.
So, before we continue, how is homologous chromosomes defined? Or how are homologous chromosomes defined?
The chromosomes are from the kuan, ma'am, from it's either from the female or from the male, ma'am. Ang chromosomes na
What is the definition of homologous chromosomes, Ms. Elnas?
Ma'am, kanang two pieces of DNA within a diploid organism. So, carrying the same genes from each parent source.
What else, Mr. Gador?
Murag same ra mi ma'am nga each carry genes controlling the same inherited characters.
Okay, what else? Mr. Soliera?
Kuan ma'am. Ang homologous chromosomes are a set of one maternal and one paternal chromosomes that pair up with each other inside a cell during fertilization.
During fertilization?
Ay, before.
Hmm?
For fertilization, ma'am. During sa process sa kuan meiosis.
It doesn't mean that they pair during fertilization, right?
Yes, ma'am.
Okay. Go on, Mr. Salapa. So what's the difference between prophase of mitosis and meiosis 1?
In meiosis one, ma'am, it's the koan. It has a homologous chromosomes which is from each maternal.
Dili lagi it has homologous chromosomes. There is pairing of homologous chromosomes, which we call a synapsis, and then exchange of genetic material between homologous chromosomes, which is or otherwise known as crossing over. Okay? Homologous chromosomes are also present in the cells that undergo mitosis. Okay? Hmm. So there is an addition of two um, remarkable events. First, synapsis followed by crossing over. Okay. Next, how about metaphase?
Metaphase, the the homologous chromosomes, it is aligned through into the equatorial in the metaphase plate. Then each spindle fibers, it is attached to the kinetochore, the ani in the kinetochore. Then mm, after during anaphase, so
Wala pa, metaphase pa ta sa lapa, metaphase pa. Asa nakakonect ang kinetochore microtubule sa metaphase sa mitosis?
sa kinetochore, ma'am.
Now, how about sa meiosis 1?
Sa meiosis 1.
Hmm.
Sa kinetochore nila, ma'am.
How is it different?
It's different because the it has a homologous chromosome. So after the pulling over is each poles will receive a one amount of chromosomes and one amount of chromosomes diri pod sa opposite pole, ma'am, which is kanang dili mabungkag ang chromosomes each because kay ang homologous pairs kay galutod man sila, ma'am. Then the attachment of kinetochore is into the homologous chromosomes, dili tungod ilahang pag-attach sa ma'am, sa spindle fiber nilang duha, ma'am. Bali magbulag sila ba having mawala na ang homologous chromosomes o homologous pair of chromosomes during metaphase plate.
Mr. Montegrejo?
Yes, ma'am.
How is the attachment of the kinetochore microtubule different in meiosis 1 metaphase versus that of metaphase of mitosis? Static ko, guys. I'm using my headset. Static akong headset. Ay naay kuan saba.
Wala ra, ma'am.
Kung headset ka gubaon. Sige. How is it different, Mr. Montegrejo?
Ang difference nila is that sa metaphase 1 of sa meiosis, ang pairs of homologs are positioned at the metaphase plate, whereas in the metaphase of mitosis, it is the individual chromosomes that are positioned in the metaphase plate.
Yeah. The positioning of the chromosomes is different. What about the kinetochore microtubule attachment?
I'm asking about the position of the or the attachment of the kinetochore microtubule. Miss Triplet?
At the centromere, ma'am.
Hmm? Are you sure it's at the centromere?
Kuan ma'am, in mitosis ma'am,
Hmm.
the attachment of the spindle fibers in kuan is kuan na siya ma'am kanang into the in one chromosomes the attachment of the spindle fibers is kanang gatap na attach sila siya sa isa ka kuan ma'am, isa ka chromosomes. While in meiosis ma'am, the attachment of kinetochores is naka-attach siya sa another chromosomes, then ang usa pod into the opposite pole nga spindle fiber is naka-attach pod siya sa homologous pair nga chromosomes ma'am.
Okay. So sa mitosis, ang imuhang both spindle microtubules are both kinetochore microtubules are attached at both or the same pair. The pair of kinetochore that is found in one chromosome. Okay. Whereas kining sa meiosis, one of the kinetochore microtubule from a separate pole is attached to one kinetochore on one of the homologs, and then the other pole or the other kinetochore microtubule found on the other pole of the cell is connected to the other homolog. Okay, so that's the difference. Para during anaphase, the homologous chromosomes are separated.
Yes ma'am.
Okay, and not the sister chromatids.
Yes ma'am.
Okay, let's go to anaphase.
So anaphase, during anaphase it's kuan sa metaphase is nahitabo during metaphase nahitabo man dre ang independent assortment ug sa prophase 1 ang the crossing over. So during anaphase, the spindle fibers attach into the kinetochore of the chromosomes, then dere na magmukuan na po siya iyahan ng birahon paduol ang kuan ang chromosomes to form birahon niya ang chromosomes.
How is it related to independent assortment, Mr. Salapa?
The independent assortment ma'am is kuan man na kanang the randomly aligning of kuan kanang chromosomes in the center or in the equator of the metaphase plate ma'am, nga which is makakosya og kuan genetic diversity ma'am.
Okay.
Then so mo na to during anaphase is mo sugod na og kuan kanang pangbirahon na siya sa kuan sa centrioles ang H kuan kanang chromosomes, then diren mo proceed na dayon sa telophase, which is the formation of kuan cleavage furrow kay mahitabo na siya, then it would divide through cytokinesis, which is magbulag na siya. Then after this na from data ko yung mga PowerPoint day ari maon siya mga kalam tara tetrad bivalent katong sa prophase pa na siya.
So what's the difference between the anaphase of mitosis and anaphase of meiosis 1?
Sa anaphase of meiosis H is nahitabo ang kuan ning ang homologous pair kay kuan kanang gi-separate siya, while sa mitosis is kuan man dito ang naa dito kay kuan man sister chromatids, which is wala man siya kuan random assortment or the exchange of kuan genetic material. So mo'y naka-difference nila ma'am. Kay kani pare diani mahitabo ba pare aning kuan green o blue na combination, which is wala siya sa anaphase in mitosis. Nahitabo siya in the anaphase 1 of meiosis ma'am.
What else?
Okay, proceed ma'am.
No, what else aside from that? Ano pa ilahang difference? Ano to? Ang sa mitosis is walay
wala na hitabo ang kuan ma'am, wala sila genetic exchange of genetic material, which is ang pag-pull over nila is kuan bali nahitabo dre na siya kuan independent assortment, while sa mitosis is kuan raman siya kanang may exact copy of the kuan raman.
Remember, I'm talking about anaphase of mitosis and meiosis. There anyone from the group? Anyone from the group?
Ay kuan ma'am, dara ay ma'am. During anaphase 1, the spindle fibers are attached to kuan chromosomes, but still the sister chromatids are kuan pa siya ma'am, is still joined together ang sister chromatids, while sa anaphase 1 in mitosis is the sister chromatids are being separated naman, bali natunga na siya.
Okay, making it full-fledged chromosomes, right?
Full-fledged chromosomes ma'am.
Okay.
So that's then after is the telophase will happen. So kuan the in telophase 1 sa telophase 1 sa meiosis is still the sister chromatids are still kuan gyapon siya ning wala pa gyapon siya natunga or intact pa gyapon siya, while then sa mitosis ang sa during telophase is na nabulag na ang sister chromatids, which is nag-distribute na yung genetic material to form the exact copy of the daughter cell, while sa meiosis is kuan ning kuan pagyapon siya kanang sister chromatids are being wala pagyapon siya nabungkag. So after the formation of cleavage 4O is
Taisa. How about the number of chromosomes?
Yes ma'am, the number of chromosomes is kuan is 46 and 46 still.
Taisa, nganong magamit man ka og chromosome number?
Ay kuan po siya, bali the number of chromatids ma'am.
Number of chromatids? Are you sure? Can you use ploidy instead of the number of chromosomes, like haploid, diploid, triploid?
Yes ma'am. So
the number or the number of sets of chromosomes instead of using
sets of chromosomes, it's still kuan diploid during telophase 1. Then after it will
diploid siya after telophase 1?
Yes ma'am, diploid pa siya ma'am.
You're sure? Diploid gihapon? Remember, I'm talking about meiosis 1.
Sa meiosis 1 ma'am?
Yes.
Sa meiosis 1 is kuan na siya, haploid na siya during meiosis 1.
How about in mitosis?
Kuan siya ma'am, kanang haploid na siya ma'am.
Mitosis.
Mitosis. Haploid na siya ma'am.
Mr. Salapa. Okay. Why is it that mitosis is considered or also known as equational division, whereas meiosis is reductional division?
Okay, because the kuan ma'am and during the production of daughter cell in mitosis produces one daughter cell having the exact copy of the mother cell, while in meiosis produces four haploid daughter cells which consist of 23 chromosomes from each.
23. And po, di ba ang ingong ko, do not use the number of chromosomes.
Yes, ma'am.
Refer to the number of set of chromosomes.
Yes.
Okay. Instead of saying 23, how do you say it?
Haploid, ma'am.
Hmm.
Yes, haploid.
So, ang product sa mitosis, diploid.
Yes,
Okay? Ang sa meiosis 1, haploid. But, unsa man? Ang sa meiosis 1.
And sa meiosis 1, it will undergo another round of
No, Mr. Salapa. It is haploid, but the chromatids are still intact.
Yes, ma'am.
Hmm.
Yes,
Another difference between the two.
The
Kapay difference nila.
are still intact.
Then, sa telophase, sa mitosis o meiosis 1, wala pa tanahuman. Ang sapay difference nila.
Another difference is in telophase, it will undergo another division, ma'am. While in mitosis, it is the end of the process, ma'am.
Deh. Harikos. Classmates, Uliara, have any idea? Aside from those mentioned by Salapa, what else? So, pa may difference between telophase of mitosis and telophase of meiosis.
Ang centromere ba to?
What about the
Each
centromeres?
each cell as in telophase 1, ang each cell nga kanang kuan sa telophase 1 will have kanang one set ba? Or tagsa-tagsa ka centromere, ma'am.
So, sa ato pa sa meiosis 1 telophase 1 of meiosis, dili na tagsag centromere?
Okay.
Matana na po kunin mo kung sa definition centromere ron. Mr. Gador. Mr. Gador?
Ma'am.
Mr. Salapa, it's almost 12. Niod to tayo, gutom na akong aspares. Mr. Salapa? Hello, Mr. Salapa. Mr. Salapa, for your assignment, Number 1 assignment, discuss the detailed process of synapses and crossing over. Katong naas book ni Hartwell, itong molecular approach. Ganahan ko makakita ato. Second assignment, Mr. Salapa, instead of pasting, you can still paste the figures, but I want you to tabulate the difference between the three processes. Katong prophase, TMAT, TMAT sa mitosis, meiosis 1, ug meiosis 2. Okay, unsa ilang mga kalahian i-tabulate. Okay. All right, it's almost 12. We have tackled several topics already. By the way, I would just like to announce, maybe we can move. If it's okay with you guys, ato lang i-move ang midterm exam. Midterm exam is supposed to be next week, May 10 to May 14 according to the academic calendar. Pero I don't think what we have discussed is enough as coverage of the midterm exam. So, is it okay if we move to the midterm exam somewhere May 17, kuan na lang siguro, May 17 to May 21. And then magama na lang tag fall if unsa inyong i-prefer May 18 ba or May 20. Okay ra? Okay
Kera,
ra?
ma'am.
Kaya ma'am.
Atong
Yes, ma'am.
i-move siya May 18 or May 20. So next week mag-discuss kaya punta. Okay? Oo. Kulang na kaayo. Supposedly before midterm na human naunta ko sa Mendelian genetics. Start na unta tag mapping. Anyway, kulang man ang time. Daghan po kayong gipatrabaho sa NORSO. Busy po kaayo. So, mo nang sige move. Okay, so we'll just have your quiz or chapter exam if we finish the discussion up until life cycles. Isa sa life cycle gani assign?
Yes, ma'am.
Okay, Elmas. Sige. Dayon ang inyong article. By the way, Miss Elmas, nag-email kasi inyong critic paper, no?
Yes, ma'am.
Can you attach it sa kuan na lang classroom para ma-trace na ko?
Wala man ko ni post, ma'am.
Nag-post ko ganinang buntag just after I saw your email.
Okay.
Okay, kindly visit the Google Classroom I posted.
Ang uban, ma'am, kay sa email or dropon sila? Or ni admin?
Wala, ikaw pa'y unang ni-pass. By the way, how about the others? Wala pa mo ni-submit sa inyong topic for research paper. Dugay na tong deadliest deadline. Murag ako na nung duman nga ni-pass kay si Tubil pa o si Triplet. Ito ba ko? Si Tubil ba ito ikaduhan?
Email ra, ma'am, ang proposed title.
Yes.
Okay. Thank you, ma'am.
Pila to, ma'am? At least 3 or mo finalize na mig usa?
You can give me 3 if you want so that I can choose a topic from that.
Okay,
And
ma'am.
then if you give only 1, then there's higher chances of getting rejected. So might as well propose 3 topics if you want one to be approved.
Sige, ma'am.
Kuan na, Mayo na. Wala pa ko nadawat. Questions before we end this call?
Katong sa article critic sa 2 ma'am. Unsa to nga article ma'am?
Katong ako ang gi-post kaning after ani katong i-present ni Silvera na to screen capture ba to ninyo ang title? Wala.
Wala
Sulat ra, ma'am.
Gisulat ra. Sige, I will post the article here sa Google Classroom. Please wait.
Sige ma'am. Thank you ma'am.
Ma'am, thank you, ma'am.
Okay. Question.
Wala ra
Another
ka
question.
exam, ma'am.
Unya nang mangumanta sa presentation, article presentation ni Saliera for this lecture. Napailang question? Ang title is
Wala.
the consequences of chromosome segregation errors in mitosis and meiosis, right? Ano sa ganito ipasa? Limo po. Wala ko nag-set of deadline.
Wala, ma'am.
Kuan na lang. Kay karon ra man ako i-post. Miss Elnas, if you want to improve your paper or if you want to change something on your critic paper, please feel free to do so, ha?
Yes, ma'am.
Okay. Sige. Today is May 6, so maybe deadline is next week, May 13. Is it okay?
Yeah.
Yes,
Okay
ma'am.
kayo, ma'am.
Yes ma'am.
Okay.
Thank you, ma'am.
Nay pangutana?
Wala
Wala
na.
na, ma'am.
Okay na, ma'am.
Wala na ma'am.
Wala na, ma'am. Kay gutom na.
Alright. Sige, maniud na ta, guys. Thank you for attending our short lecture. I hope you learned something. Safe everyone, we now leave this call. Thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you ma'am. Goodbye ma'am.
Bye, ma'am.
Bye.